Let's start this topic with the plant body axis, because that is where the whole story of stems and leaves starts. A stem is not just a green stalk, it is the main supporting axis that carries nodes, internodes, buds, and leaves. While the leaf is the primary photosynthetic organ built around the lamina, petiole, and base. The key idea to hold here is that structure and function are tightly linked. The stem lifts, supports, and transports, while the leaf captures light and manages exchange. A neat exam point is to remember that many modified structures still trace back to these same basic origins. A true stem is identified by the presence of nodes and internodes, and that is the most reliable anatomical proof. Terminal buds continue upward growth, axillary buds sit in the leaf axils, and the plumule gives rise to the shoot system in the young plant. Even if a structure grows underground, it can still be a stem if these features are present. That is a classic trap in exams, especially when tubers or rhizomes are involved. So always check the origin and the node-internode pattern first. The stem does far more than simply hold the plant upright. It spreads out branches to position leaves, flowers, and fruits for better exposure, conducts water and minerals upward, and also moves prepared food through the plant. Once you move beyond the ordinary role, the stem begins to show upgrades like storage, support, and vegetative propagation. That is the larger evolutionary idea here. A stem can become specialized without losing its identity. The most important habit is to ask, what is the basic stem function and what is the modification? Underground stems are a perfect example of modification for survival. Potato is a tuber with eyes, which are actually axillary buds at the nodes, so it is definitely a stem and not a root. Colocasia forms a corm, ginger is a rhizome with horizontal growth, and turmeric and zaminkand are also underground stem forms that help the plant survive unfavorable conditions. The trap here is very common. Students often memorize potato as a root because it stays underground. But the presence of buds and nodes gives away its true nature. Aerial stem modifications show how the axillary bud can be transformed for different jobs. In climbers like gourds and grapes, the stem becomes slender and spirally coiled, serving as a tendril for support. In defense forms like citrus and bougainvillea, the stem becomes woody, straight and pointed, functioning as a thorn. Both structures arise from the same source, which is why origin matters more than function alone. That distinction is often tested, so do not confuse a stem tendril with a leaf tendril just because both help in climbing. In harsh environments, stems can even take over the job of leaves. In arid plants like opuntia, the stem becomes flattened and photosynthetic, while in euphorbia, it may remain fleshy and cylindrical. This is a beautiful example of adaptation. When leaves are reduced to avoid water loss, the stem assumes chlorophyll duties. The lower half of the slide shows stem spreaders used in vegetative propagation, such as underground runners, aquatic rosettes, aerial arching, and oblique basal forms. These are all clever strategies for multiplication and colonization, not just random growth patterns. Leaf anatomy begins with the shoot-apical meristem and the order of leaf development. The leaf base anchors the leaf, the petiole acts like a flexible stalk, and the lamina is the broad green blade where most photosynthesis happens. Venation and vein architecture inside the lamina are essential because they support transport and rigidity. One high-yield point here is that axillary buds occur in the axil of both simple and compound leaves, but never in the axil of leaflets. That is a favorite exam trap, so keep that distinction sharp. Leaf-based specializations are easy to remember once you connect them to plant family patterns. In monocotyledons, the leaf base forms a sheath and partly or fully wraps around the stem. In legumes, the leaf base becomes swollen into a pulvinus, which acts like a flexible hinge and helps with movement. These are small features, but they are very useful in identifying plant groups. The larger idea is that the leaf base can help with support, flexibility, and protection, not just attachment. Venation is one of the simplest ways to classify leaves by internal transport pattern. In reticulate venation, the veins form an interconnected network which is typical of dicots. In parallel venation, the veins run side by side, which is typical of monocots. This is such a high-yield clue that it often appears in direct identification questions. If the venation pattern is clear, classification becomes much faster and much less confusing. The incision rule is a very practical way to separate simple and compound leaves. If the incisions do not touch the midrib, the leaf is simple. If the cuts reach the midrib and divide the blade into separate leaflets, the leaf is compound. This single rule removes a lot of ambiguity, especially in visually confusing specimens. In exams, the safest approach is to trace the lamina carefully rather than guessing from the outer shape alone. Compound leaves come in two major forms. In pinnately compound leaves, the leaflets are arranged along the central axis called the rachis, as in neem. In palmately compound leaves, the leaflets arise from a single point, usually the tip of the petiole, as in silk cotton. The visual difference is easy once you think of a feather-like layout versus a hand-like layout. That mental picture helps far more than memorizing names in isolation. Phyllotaxy is simply the arrangement of leaves on the stem axis, but it is one of the cleanest identification tools in botany. Alternate phyllotaxy places a single leaf at each node. Opposite places a pair of leaves at each node, and whorled arrangement has more than two leaves at a node. These patterns are not just decorative, they help optimize light capture and reduce crowding. A quick association like China rose, mustard, and sunflower for alternate can make recall much easier in revision. Leaves can also be modified for functions beyond photosynthesis, and that is where adaptation becomes especially interesting. Tendrils support climbing, as in pea. Fleshy leaves store food, as in onion and garlic. Some petioles become photosynthetic, as in Australian acacia, and spines protect plants from herbivores, as in cacti and insectivorous plants. The exam habit to build here is to ask whether the modification involves support, storage, synthesis, or defense. That one question usually leads you to the right answer quickly. This is the trap slide, and it deserves careful attention. Stem tendrils in gourds and grapevine arise from the axillary bud, while leaf tendrils in peas arise from a modified leaf part. Stem thorns in citrus and bougainvillea also come from axillary buds, whereas leaf spines in cacti come from the modified lamina. The real test is not what the structure does, but where it comes from. If you memorize origins instead of just functions, you avoid a huge number of mistakes. By the end of this chapter, the plant axis should feel like a clear blueprint. Stem means nodes, internodes, and buds. Leaf means base, petiole, and lamina. And each of them can be modified for survival, support, storage, or reproduction. The stem builds the framework of the shoot system, while the leaf handles light, transport, and adaptation in elegant ways. If you can recognize the origin of each modification, the topic stops being memorization and becomes logic. That is exactly the kind of understanding that makes you ready to tackle any problem.